Good morning, good afternoon, and good evening, everyone, and welcome to another edition of JAX Live. I'm your host, Danielle Fontaine, a senior analyst with the Strategic Insight and Innovation team here at the Jackson Laboratory. And today I'm pleased to introduce Dr. Michael Ellis, Associate Director with Product Management and Business Analytics. As a reminder to our audience, please be sure to use the comment box to submit any questions you might have, any comments you might have, or just a shout out from wherever you're watching from today. We'll answer as many of these questions as possible at the end of our discussion. So to start us off, today's topic is JAX innovation, where we see preclinical research going. We know that the field of preclinical research has generated some incredibly impactful results over the past decade, but has also faced some pretty significant challenges. So Michael, my first question for you is, can you speak to some of these challenges and any shifts that you see happening in the field that are poised to address them?
Yeah, sure. Thanks, Danielle. It's good to be here with you. It's a bit of a shame that we have to get on a live stream just to have some time to chat. But you asked about challenges in preclinical research, and I think that pretty much anyone in the space would agree that despite some really significant contributions to medical science, that there are some real major shortcomings in the space that I think we as a community can collectively overcome. If you sort of look back in history, animal research, and particularly research using mouse models, has played an essential role in just about everything from the polio vaccine all the way up to modern gene therapies like the muscular dystrophy therapies that are getting a lot of press lately in clinical trials. Yet the success rate for all of this work is not nearly as good as it could or should be. Depending on which analysis you happen to read, the statistics on translatability vary anywhere from about a third all the way down to less than 10%. And there's a lot of possible reasons for why this would be. Some of those could be based on study design. Often these studies can be underpowered. Some of them could be based on a true lack of conservation between animal model and human. But I believe that a significant portion of it could be attributed to the use of less than ideal models. And the good news is that that can be addressed by some technology platforms that are available today. And there's three particular technology platforms that come to mind that I think really have the potential to shift the translation fraction between preclinical and clinical. The first of those is the increasing focus on the use of advanced model generation tools, in particular to create complex constructs to model disease or potentially even the underlying biological mechanisms. The second of those is the increasing recognition within the scientific community of the importance of genetic diversity in animal research. And then finally, the third is the increasing focus within the community on the use of humanized mouse models to better understand human biological mechanisms.
Great. And so to get to your point about the different technology shifts that we've really seen in this preclinical space, what's the impact of some of those genetically unique models?
Yeah, so we've continued to see an increasing focus on the use of complex models of disease, largely due to the ubiquitous availability of advanced model generation tools. And I think that sort of the natural reaction in this space is, yeah, that's not new. This has been around a long time. CRISPR isn't even new anymore. Even the Jackson Laboratory itself has more than 11,000 unique mouse models within its library. And there's plenty of other biorepositories around the world with other models in them, both mice and other species. Despite all of that, I think we are still at the beginning of a shift towards precision models for specific research questions. If you actually look at the vast majority of the preclinical research that's happening, it still happens on a very small subset of models. Oftentimes, those are inbred mouse models or particular models that have been made decades ago. In particular, the metabolic space comes to mind in thinking about a domain that uses a lot of models that have been around for a really long time and aren't that great in terms of recapitulating the human disease. And yet, we're already starting to see a shift towards sort of more of these precision models away from those legacy platforms towards those 11,000 models that are in our library. As well as some new model creation as researchers are realizing the translational potential of these precision models.
That's great. And so you've mentioned new model creation. Are there some shifts in how researchers are really approaching creating those new models?
Yeah, and this is something that I think Bill Buis and Rob Taft and Don Tanamachi covered in last week's live stream, which if folks haven't had a chance to see it, they can go onto JAX's YouTube channel to see some of these past live streams. And they've definitely done a better job of talking through this more eloquently than I could. So I'd encourage you to go watch those to get a better view of that. But we've seen an increasing recognition with our customers and researchers that we work with of the importance of genetic background in terms of making new models and the function of genetic background on the ability for those models to provide translational relevance. Researchers are definitely getting more and more savvy about how to build complex models and the fact that building that model on the wrong background can lead to either a non-functional model or potentially worse, a model that's functional but that has impacted translational relevance. And typically that means you won't discover the issue until many years later and potentially millions of dollars later. As always, Bill has done a much better job of covering this than I have. I'd encourage folks to watch that live stream. And I'd also encourage folks to give our folks at the Technical Information Science Group a call if you have any questions about the role of genetic background in model generation. They can either help you directly or can put you in touch with the appropriate expert.
Oh, definitely, definitely. We know that genetic background, as you mentioned, plays a really important role sometimes in translatability. But what do you see as the importance of incorporating diversity into your preclinical research plans?
Yeah, that's a great question. The importance of genetic diversity in preclinical research has been pretty well understood and demonstrated for well over a decade at this point. But more recently, we've seen an inflection point in the recognition and interest in these platforms, particularly in the last year or two, as more and more of the researchers that we work with have approached us with questions about how to incorporate genetic diversity into their research. If you actually think about it, research based on a single inbred mouse strain is pretty much analogous to research based on a single human patient. And it has very limited potential for broader understanding or inference. I don't think anyone would suggest with a straight face that we conduct clinical trials with just a single patient. And yet, we've been doing pretty much exactly that in the preclinical space for decades without much reservation. Don't get me wrong, Danielle, there's tremendous value in deploying genetically identical mice in order to validate our understanding of a biological mechanism. But it's really just as important to go broad and to evaluate across a genetically diverse population in order to understand if your discovery is unique to the mouse or if it's applicable to a broader population. And I don't think that most folks would want the first in human to be the time where they're discovering the impact of genetic diversity on their therapeutic, if that's what they happen to be working on. I'll say that this is definitely especially important when we're talking about complex multi-gene diseases where larger variability between individuals is expected. And I'll also add, Danielle, that there's a lot of publications that demonstrate the use of these diversity models in different domains and how these diversity platforms have led to novel conclusions that very likely could not have been reached with classic inbred models.
Yeah, so just for anybody who might not be familiar with genetic diversity, when you talk about these diversity models, are you talking about outbred strains?
Yeah, good point. I should clarify. So when I'm talking about diversity models, I'm referring to models that possess internally diverse gene pools, which can include traditional outbred models, as well as more sophisticated models like diversity outbred or the collaborative cross mouse. The problem with these traditional outbreds is that they've proven to be somewhat lacking in genetic diversity, and whatever diversity they do have is relatively poorly controlled. Whereas the diversity outbred and the collaborative cross mouse, these have been developed explicitly to maximize genetic diversity and have well-defined genetic inputs, which can be extremely beneficial. I will say that this is true for myself and many others. It can be pretty intimidating to try and understand these platforms and to dive into figure out how to use them. Generally speaking, the Jackson Laboratory has some pretty good resources to help folks through this. I'll point to the Jackson Laboratory's website on genetic diversity, which points to a lot of different papers published using these platforms and talks through when and where you would use a particular mouse for a particular purpose. And I would also suggest that there is a previous live stream in which one of the folks from the Jackson Laboratory talks through these platforms. The last thing I would add to that, Danielle, is there's a number of use cases that for genetically diverse mice, but to keep things relatively simple, I think there's two in particular that are pretty easy to get into. And that's the space where we've seen a relatively large inflection point in interest from our customers. The first use case is really simple. It's just about adding a diversity arm within your research plan. And there are some really nice papers out there that are linked to on the JAX diversity website that demonstrate how a relatively small cohort of diversity outbred mice in a single arm of a study can be used to provide important learnings and redirect future work. And in often cases, this is just a single figure within the context of a greater paper. The second use case is drug safety screenings, where population dynamics are a pretty key measure of overall safety. And not only can you screen across a wide genetic pool with these platforms, but you can also, if necessary, dive in to understand what are the genetic drivers of response.
Oh, that's great. And so just in the interest of time, moving on to one of the other platforms that you talked about a bit earlier, what do you see as the impact of using humanized mice in these preclinical platforms?
Yeah, that's an interesting question. And I'd say that the answer to it is pretty rapidly changing as humanized mice make their way into increasingly diverse applications. I'd say maybe before we dive into that, though, I should clarify, Danielle, that when I'm talking about humanized mice in this particular setting, often that term can be used to apply to both genetic and cellular approaches, but I'm particularly focused on the cellular approach, where you're talking about engraftment of hematopoietic stem cells into an immune deficient mouse in order to provide it with an immune system that's more analogous to that of a human. In that particular setting, the folks that have been working in the infectious disease space as well as the oncology space are likely pretty aware of these platforms and how they can better model the human immune system and the interactions of it with an infectious agent or with a tumor. But one of the big shifts that we've been seeing is the increasing application of these models in other domains that are well beyond those sort of traditional stomping grounds of these platforms. Pretty much any application that has an inflammation-driven disease process is applicable and can benefit from these models. And I would say that more and more folks recognize that inflammation is a contributor to a wide range of diseases, including metabolic disease, cardiovascular disease, even neurodegenerative disease like Alzheimer's. And we at the Jackson Laboratory have seen a pretty big uptick in interest and utilization of these humanized mice in those areas as well.
So that's great. So do you expect these humanized models to be increasingly used in things like efficacy studies for all of the areas that you just mentioned?
Yeah, absolutely. But I wouldn't say that it's limited to just efficacy studies. One of the areas that we're gaining a lot of attention and interest in more recently is the application of humanized mice for safety evaluations, in particular of immunomodulating therapies like bispecifics or CAR T therapeutics. These therapies have a particularly greater risk of causing cytokine release syndrome, which is pretty much the primary cause of their failure in the clinic, or one of the primary causes of failure in the clinic. And that's an issue that I think was first highlighted back in 2006 by the TeGenero 1412 trial, which led to some severe morbidity that was precipitated by cytokine release syndrome following the infusion of the drug in that trial. But even today, it still plagues many immunomodulating therapeutics. I think as recently in the news, I think Poseida's CAR T therapy was subject to some of the same challenges within the clinic. Classic in vitro assays and animal models have pretty much failed to predict these safety issues in these immunomodulating therapies up to this point. And there's now really good data, though, suggesting that humanized NSG mice can recapitulate the cytokine release within an in vivo setting, and they can do so with really good sensitivity and specificity. And as a result, we've actually been getting a fair number of calls about this application, which has been increasing more recently. And I wouldn't be surprised to see most immunomodulating therapies tested on this platform before too long, especially given how serious clinical failures can be with these therapeutics.
Oh, that definitely makes sense. And unfortunately with that, we are just about out of time. So Michael, do you have any kind of closing thoughts on this topic before we go?
Yeah, I do, Danielle, and thank you for having me. You started this off asking questions around some of the challenges with preclinical research. We touched on a couple of different technology platforms. Obviously, in order to bring preclinical research to become more and more translatable into the clinic, there's a number of different things that have to happen in sequence. And I sort of touched on some of those in the beginning. But these three technology platforms that I'm referring to here, I think are really key in driving preclinical translation into the clinic and are really important. And I think we're just at the cusp of seeing folks use these platforms more and more to drive that translational relevance.
Great, that's very exciting. So thank you for everyone for joining us today. If we couldn't answer your question, we will be sure to follow up with you. And if you have any questions about anything we've talked about today, please reach out to us at micetech@jax.org or look on our YouTube channel for other live streams that we've mentioned during this presentation. So again, thank you for joining us, and thank you so much, Michael, for the